So my name is Ben Twining, and I live in Brunswick, Maine, and I work at the Bigalow Laboratory for Ocean Sciences, which is in Boothbay, Maine. So I'm an oceanographer. I think of myself as a marine biogeochemist, to be a little more specific. But I study what the water is made of and what the things that live in the water are made of. So one of the reasons I became an oceanographer, there's a couple of reasons. Two of the reasons I became an oceanographer were the chance to go to sea and to do projects like this, where you get to be surrounded by your colleagues for a month or two, and you get to just like immerse yourself in science really around the clock, which is really fun. I know that's going to sound really nerdy, but it's really true. But the chance to travel, we get to go to some pretty exotic places, and we get to see some things that are really unique, right? We're probably going to get to see some icebergs on this cruise, and that's really cool. So I love that part of oceanography. I also really like the team part of it. So oceanography is really a team sport. It's really rare to have someone do a project by themselves. We're out here with 37 scientists, and we need all of 37 of those people to make this work. And I really enjoy working with other people. I enjoy sort of pulling us all together to work towards something that's bigger than any one project we're doing. And I love that about oceanography. And I'd say environmental science in general is often, because you have to look at so many different aspects of the environment in order to understand it, you usually need a team. And I really like working with a lot of other people on that. So on this cruise, I'm the chief scientist. So I'm in charge of just organizing everybody, making sure everybody knows where to be, what they need to bring, making sure that everybody's science fits together and that everybody has the resources they need to do it. So this cruise is part of the GEOTRACES program. And this is a program to figure out the concentrations, the amounts of trace elements. So these are elements that are in really, really small abundance in all over the global ocean. And so there's a couple reasons why we care about these elements. So elements is a pretty vague word. So mostly we mean metals. So metals like iron and zinc and manganese and copper and nickel, all of those are on the back of the cereal box. When you look at the cereal box, your required nutrients and minerals, some of those are metals, and we need them for the proper functioning of our body. And so we're interested in the distributions of these over the globe to understand how they control productivity, like I've talked a little bit about. We're also interested because they can be pollutants. So something like mercury, which is released to the atmosphere when we burn coal and other fossil fuels. And mercury can get into the ocean, and it can accumulate into organisms, and it can be harmful to humans if we eat those organisms. So we're studying pollutants like mercury and lead. And then another reason we're interested in understanding the distributions is because metals and trace elements can serve as clues to what the Earth was like in the past. So because they're still around and they stay around for a long time, we can measure the distributions of metals like in seawater and in different parts of the ocean, and also in the sediments at the bottom of the ocean. And we can understand what the Earth used to be like. And that's really valuable as our climate starts to change, to understand what it used to be like and what controlled it back then.
What is the deepest depth we're sampling from?
So we've so far taken some water from about 5,500 meters, so 5,500 meters, and every meter is like 3 feet. So that's 15,000 feet. That's more than 3 miles. So it's amazing how deep the ocean is. So these cruises are interesting. We collect a lot of samples, and most of them we don't analyze on the ship. Right? So even though we have a fair amount of equipment on the ship, but a lot of what we're doing is bringing home water. But there are people who are working really hard to make measurements on the ship. And so they've made measurements showing us just how low the nutrients are in this desert I talked about, this gyre. It's really amazing. I mean, the nutrients are even lower than we thought, which has been interesting. Another thing we're seeing is that we're seeing more productivity in this part of the ocean than I was expecting. And I think a little bit that shows us that the ocean is always changing, right? It's never staying exactly the same. So we need to think about where we are in the season. Like we're just sort of starting the spring, summer period down here in the Southern Hemisphere. And so I think we're probably already seeing the bloom that comes in the spring. It's probably already arrived, which is totally normal. But I think we're seeing sort of the result of that because we're seeing a lot of things in the ocean right now.
The ocean blooms in the spring.
It does. It blooms just like flowers in your garden. The ocean blooms. And so we actually timed this cruise. We left Tahiti so that we could get down here sort of right after it was blooming, We couldn't come too early because it's covered in ice, and we couldn't come too late because we'd missed the bloom. So we've tried to time it, even though it's over the holidays. The reason we've done it is so that it is just when we're catching the bloom. Most of the things that live in the ocean are really small. They're little single-celled organisms. So we collect them onto filters, and we bring those filters back to the lab, and we turn them into liquid. We digest them, we call it, and we analyze all the elements that are contained in there. And so in addition to doing that with all the stuff collected on filters, I also collect the individual organisms themselves, and I look at them with an X-ray microscope, which is really cool, and I scan them with this really bright, teeny little X-ray beam, and it causes them to glow, and each element in them glows a different color. And I can measure what they're made of by hitting them with this X-ray microscope. So I'm collecting cells out here that I can take home and figure out what they're made of. Yeah, so a big question in oceanography is what controls the productivity of the ocean? You know, how much plant material is being produced and how fast are they taking carbon dioxide from the atmosphere and turning it into plant material? That's what we call productivity. And so that changes, that limiting nutrient changes in different parts of the ocean. Yeah. So like if you're trying to build something, like you're building a birdhouse, right? You need wood and you need nails and you need a hammer. And you can't do that unless you have all three things. And similarly, if you're going to build a cell or an organism that lives in the ocean, you need all the pieces of that organism, including all the different nutrients that are used to build that. And so a really important question for us is what is the limiting material that is keeping organisms from growing as fast as they might out here? And sometimes those materials are metals, which is what GEOTRACES is studying. So one of the ways we figure out what they're missing is that we ask them by looking at their genes. And we sequence their RNA and we see what they're stressed by. And one of the things we're doing on the cruise is collecting samples for DNA and RNA measurements of the life that lives in the ocean. So we're going to send those back to a collaborator, and they are going to extract the DNA from the material, and they're going to sequence it. And with that, they're going to know basically who's out here, what organisms are living in these waters. And they're also going to sequence the RNA, and that's going to tell them how the plankton are feeling. So it's really important for us to understand not only what's here, but are they healthy? And are they growing rapidly? Or are they short of something? And we do that by looking at their RNA.